And so what people did was start looking at covariation or co-linkage of individual traits between different people in the human population. How often do you have brown eyes and, red hair? Are those ever linked? Are they not linked? Do they follow Mendelian genetics or do they follow non-Mendelian genetics? And we had, at the time, a huge number of markers that people had already decided what chromosome they were on and where they were with respect to one another on that chromosome simply by linkage analysis of the human population. And it was something that when they put together the human genome, they constantly referenced to make sure they were putting it together correctly, because they knew the genetics had to be correct, and the sequencing technologies were actually the risky part of doing the human genome. And so they used the physical markers to actually figure out if the genome was correct, and that was one of the major metrics when they actually published the sequence that convinced those of us in the field who were really interested in it that they had done it right. What's the other advantage of it? And I'll spend some time on these slides because it will be important for both your exam and for your life. How many are in this room are evolutionary successes? All right, put your hand down unless you have produced an offspring that is carrying your DNA. You all are evolutionarily worthless. It's true. You could have. Do you have the greatest DNA? Maybe. Absolutely. This gentleman right here is the greatest DNA. Does anybody else have your DNA? No. Then your DNA dies with you, and your great DNA is dead, 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 dead. So, Louisa, you bred? Woo! We got two of us in the room who are evolutionary successes. The rest of you, from an evolutionary perspective, who? No. Now, if you decide to breed and you have a child, there is some probability that that child will have some sort of non-perfect DNA like this gentleman's is right up front here. And in fact, in many families, you'll see that diseases that have a genetic basis to them will follow a family around. And we want to find out what's the underlying basis of that genetic disease and work on therapies that might be able to help them. So if a whole family is suffering from a particular disease and one member of the family isn't, then you sort of have a creative way of thinking. Well, if I can find out what's different about the DNA of the one kid who doesn't have anything wrong with them phenotypically, and the other ones who are genotypically and phenotypically have something wrong with them, we can go on it. Now, you, right, you're probably the perfect person for this family, and you two in the back, right? You've got 8 siblings, right? This makes it really convenient. We've got a lot of genetic history to go on. And in places, anybody here from Iceland? Genetically worthless and no one from Iceland. And no anthropology. They've got really great historical family histories of who bred with who in Iceland, and they can actually go and get a lot of the DNA out of these people. So let's take the example that's on the top. Both mom and dad are mating. That's a good thing. The mom actually has a disease. Mom's chromosomes are shown as, the little sticks on the left. Dad's chromosomes are on the right. And in this scenario, the mom has got a problem. She's got a gene causing a disease. Now, from this picture, we know where it is. The point is that we don't know where it is, right? It's that little red line. There's something wrong with the DNA there. If we wanted to find out where it was, and we knew where other markers were on the genome, and we knew that from all of these markers that we had identified previously before even the human genome came out, we could decide how often all of the offspring of mom and dad have both the physical marker in blue and the disease in red. And if we look at many, many such physical markers, not just the blue one, but imagine there was a green one up there. Maybe it was perfect Mendelian with the green one. Okay, they're probably on different chromosomes. But it's not perfectly Mendelian with the blue one. Well, it's probably on the same chromosome. The more and more it's, going into a 50-50 split, the more likely it is that red and blue are co-segregating. So if you look at the 7 kids down here, 4 of them have the disease. Of those 4 that have the disease, 3 of them also carry the marker. 3 of the kids don't have the disease, and none of them have the marker.